Okay, it's 4:01. So I'm going to start talking. We still have the numbers going up a little bit, but I'd like to welcome everyone. Thank you for joining our Nantucket Field Station Spring Seminar once again. And today I am thrilled to be introducing a fellow UMass person. Before I get to that, I would just like to say, if you're not aware, my name is Yvonne Valancourt. I am the director of the Nantucket Field Station, and we are a facility of UMass Boston, managed by the School for the Environment. And we sit out here on Nantucket Island on over 100 acres, owned by the Conservation Foundation, Nantucket Conservation Foundation. And it is a beautiful spot. We support courses, which we're about to start our summer courses this weekend. And we also host quite a range of researchers, not only from UMass, but from a variety of agencies and institutions. And before we get into our talk, I would just like to point out that this is a webinar format. So we will be hearing from Dr. Jeremy Anderson. And at the conclusion of his talk, we will entertain questions, and you can record the question as they come into your head. If you like, there is a question and answer function button at the bottom of your window. So if you look to the bottom, you'll see Q&A, and just click on that and type in your question, and we will try to answer as many as we can. So without any further ado, it is my distinct pleasure to introduce Jeremy Anderson, who is from UMass Amherst, and he is in the Department of Environmental Conservation as well. And he's going to tell us all about invasive knotweed, which is quite a problem, and that's why so many of you are tuning in. And he's also going to tell us about different control management practices, and that includes biological controls. So I will let him take it away. He can share his slides and unmute himself, and I will mute myself. So once again, thanks everyone.
And there, did it work? All right, excellent. Thank you so much, Yvonne. And I am very thrilled to share with you the work that we're doing on the biological control of Japanese knotweed. Also, just to talk about knotweed, because it's, although it's a horribly invasive plant, it's a really cool plant. And it has done a really good job of becoming, as what it is today. So this beautiful critter, this is Aphalara itadori. It's a psyllid, and we'll talk about that at the end. But first, let's just jump into what is knotweed. One of the first things when we talk about knotweeds is to say what it is not. And it is not a bamboo. Many people think it's a bamboo, which is a monocot, so a grass, but it's actually a dicot. It's an angiosperm. It's a flowering plant that you can see in the photo here, these beautiful floral structures. And after that, it gets a little funny. And it's a species that, even though it's so common and so well studied, there's a lot of disagreement of what it is. We talk a lot in science, in biology about, if we can't name something, then how do we know what it is? And knotweeds are in that kind of genre where we don't, we keep going back and forth, we as in the people who study the angiosperm phylogeny, about what should we call it? Right now, it's being called Reynoutria for the genus, but historically it's been Fallopian, it's been Polygonum, even for Japanese knotweed, the species name keeps changing. So a lot of confusion, which kind of makes studying the biology of it so much more difficult, because you're like, what is it? And that's always a problem. But for some reason, the common names have stayed the same. And that's usually we talk about like, oh, you use the scientific name because that clears up the situation. Here, we want to use the common names actually help. So we've got Japanese knotweed, which is common as most of Eastern North America. We have Japanese as the dominant species. Giant knotweed, which is much larger, and I'll go over some characters next, which is present, but primarily is the dominant species on the West Coast. And then we have Bohemian knotweed, which is a hybrid between the two, and that can be found in both locations. So all three species, they can be present, and they can be in any location, really. So it becomes, once you get to your location, what do you have? And kind of the first pass, the easiest thing to do is to look at the size of the leaves. Giant knotweed has these beautiful heart-shaped leaves. They can be 8 to 16 inches in size, so that's a considerably large leaf. The fluorescence can be different, kind of more compact and upright. And generally, and again, with plants, it's hard to have like a perfect, this is where it's always going to be. But generally, you have more of kind of a vertically straight up and down plant. It's not always true, and sometimes you might see that for any of these species a little bit of overlap. Japanese knotweed, which is what we have here in Western Massachusetts, is the dominant species. It's a much smaller leaf, and it's a much like kind of the base. If you think of a shield, that's kind of the shape of the leaf that you're looking for. There's almost flat, although you will see a little bit of heart shaped, very most of the time, but much smaller leaf, and also you have these kind of like droopy fluorescences with a more zigzag structure for the plant itself. So if you're looking at last year's growth, you can picture it being this brown and kind of it's deceased, but you can see the zigzag in the stand. Bohemian's a little more difficult because it is a hybrid of the two species. So it's a larger leaf than Japanese, but there is a lot of overlap. And it's a little, it's flat-ish, but it does have a little bit of the heart shape at the bottom. So generally, if it's a larger leaf, if it's got the fluorescence and kind of like a mixture in the, not quite zigzaggy, not quite straight, it's likely Bohemian. Sometimes the bottoms of the leaves can have hairs, which can be a character to differentiate them, but it's tricky, and that makes, again, studying these species a little bit more difficult. So where are they from? Well, knotweeds, the genus of knotweeds, this is a kind of northern hemisphere species for the most part. These are records from GBIF, which is, if you're unfamiliar with GBIF, it's an amazing resource that you can put any species in, and it gives you natural history collection information. People who use iNaturalist, it goes right into GBIF, so you can look at the distributions of species. You can look at it through time. You can look at it, by month. And so if you are interested in the biology of any organism, I'd highly recommend going here and just learning what you can through it. So right now, as of a few days ago, there were over 320,000 records of knotweeds. But for the invasive species that we have, they're from East Asia. So these are from a natural distribution of Japan, China, the Koreas, and into kind of the Russian islands up north of Japan. And from there, they've been spread all around the world and become the invasive species that we know and love, I guess, or try to deal with it. But so how did they get here? These were really early introductions from Japan. And this is not, a lot of the pests and weeds that we have today are accidentally introduced. They might come in on shipping containers. But right now, but this species, this was one that was intentionally introduced. It was a kind of promoted as a prized landscape plant due to its ability to rapidly grow. In the stands that you probably see on the island, the stands that we have here in Western Mass, they can be more than 6 feet tall. They can be even taller than that if it's giant knotweed. So, this is a fast-growing, very tall plant, and so it was promoted as for privacy screens. A lot of the other characters that we now think of as why it's so invasive were considered good. There's very few diseases known from it. There are very few herbivores. Very low maintenance plant. We grow it in the greenhouse, and it's wonderful. If you forget to water it for a week, it just keeps going. It grows in any habitat. I'll show you pictures of it coming through asphalt. And this was incorrect, but it was thought that it had this dense underground root system that could be used for erosion control. So maybe the sides of the roads or even riparian areas, it was kind of promoted to help shore up the erosion. And then another character, which is mostly for Japanese knotweed, it was thought that this was an asexual plant. And at the time, especially in the late 1800s, a lot of the invasive plants that we have, or invasive weeds, were asexual, or they were promoted. They're like, oh, look, this is great. This is an asexual reproducing plant. We don't have to worry about its seeds being dispersed by birds and it becoming a problem. But they didn't think about, well, you just need one. And then once it's there, it's just bloop, 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 bloop. It's going to take over. So unfortunately, a lot of the characters in the 1860s that were thought to be really wonderful today are what we're fighting against. And so how does it spread? Well, not that long ago, actually, we thought it was almost entirely one clone for Japanese knotweed in Eastern North America. But that's really changed with the genomic revolution, people being able to get genomes of plants like knotweed. And as the more and more people, scientists look at the genetic diversity of knotweeds, the more that it's just, it's amazing. There's a lot of genetic diversity within all three species, Japanese knotweed and Bohemian knotweed, which were thought to reproduce primarily asexually, which you first have to say, well, how does the hybrid species form then if it's just asexual reproduction? But that's besides the point. 93% or more of the seeds are viable, and almost every Japanese knotweed plant makes seeds. So there's an incredible potential for sexual reproduction, even though it kind of wasn't acknowledged at first. And so you can have limited dispersal from the seeds in the region, but also there is a lot of asexual reproduction that's occurring from the rhizomes as the plants spread out, and that's why it makes those dense mats. And that's what you see here. This is from the Catskills. This is a riparian area where knotweed has just completely taken over. And on one hand, it's very lush. It does grow very well, and so a lot of times people say, well, what's the problem? I mean, it's not the worst looking plant ever, but it really changes the ecosystem. Now that's when there's a lot of discussion right now about what to do about invasive species. And the problem with invasive species like knotweeds is that they are so dramatically changing the community of plants and animals that coexist with them that we can't not do something about it. The consequences are pretty dramatic. So this is from a new paper published by Arnold and Tran in 2018 that really shows that once you get knotweed stands building up in a river system, it just changes the downstream flow. For rivers, we talk about river meandering, where as the water is coming down, it's always moving the river banks. But because knotweed is such a poor, it's just such a poor job of shoring up the riverbanks, it really is channelizing rivers. It's making the speed much faster, and it's changing the oxygen level in the stream, which results in fewer invertebrates. If you want to fish, if you're a fly fisher person, then that's a real problem, because now you've got fewer fish. And so that's a lot of the concern from anglers is coming from the lack of fish in knotweed infested areas. And this photo on the bottom right, that's from Vermont. But if you were in Western Mass, that photo is from the fall, because you can see the floral structures. But that's what the Deerfield River looks like right now in Western Mass. It is pretty much from this time you can drive along it to where it goes up into the Berkshires. It's just a continuous stand of knotweed. And it's amazing how quickly it can take over and change an ecosystem. In addition, not just in riparian areas, it's a problem because it outcompetes native plants. So if in anywhere, when you see knotweed coming up, you'll start to notice that it's the only thing coming up. And when there are other species, they might not be the ones you want. So this reduced diversity means that there's fewer invertebrates again. There's fewer vertebrates. This has been linked to declines in amphibians, mammals, and unfortunately for the work that we do, when you go there, there's a lot of things you don't want. Oriental bittersweet seems to do really well, having these disturbed understory where the knotweed is present. Poison ivy really loves it. So when you start walking through a knotweed stand, you're just walking through poison ivy. And this is just personal anecdote from doing field work. It is amazing deer tick habitat. We have these white no-see-um mesh bags that I'll show you in a couple slides, and it's horrible. Like you put them down on the knotweed plant and you take them off, and they're covered in deer ticks. So that's in Amherst. I don't even want to know what it's like on Nantucket, and yeah, just take a moment. Okay, moving on. Thankfully, this is not a problem that we have in the United States, but this is a problem if you lived in the UK, where homeowners are required to remove knotweed from their properties prior to sale. So this is from Newsweek. I mean, this is an older, it's 2014. But so they call it the invasive plant that eats the value of your house, because you're talking about remediation up to 4 feet of topsoil being removed before you can sell your house. And that's, you know, for some people, that's more expensive than their house, although maybe not today. But so what do we do about it? Well, we've got a few options for controlling knotweed. The first is chemical control. It is a species that does respond well to chemical control. The problem with chemical control is that it has to be the right control. It has to be done at the right time. And it has to be a chemical that's approved for where you are, depending on where you might be. A lot of times knotweed's associated with wetland habitats. So that's something that you want to make sure that if you are doing the chemical applications, that you have, first you have the appropriate permits for working in the location you're working in. You have the appropriate safety gear, as this person does here, and that you're kind of making sure that the chemicals aren't going into an aquatic or wetland ecosystem. And I do have some slides at the end, or links at least to recommended chemical controls that you can get from UMass Extension. The other technique that a lot of people have been embracing, though there's a few different variations on this, there are mechanical controls. So something that does seem to be quite popular right now is on the slide on the left, which is to put down a mesh. This really only works for a small stand, but if you can think about it, if you put down a mesh, kind of like a thin rat wire or similar, it'll girdle the plants as they're coming up. So you could put this down, you could kill the knotweed plants as it's coming up, or at least the shoots, and then let the other plants, maybe grasses, grow through and kind of outcompete the knotweed in the process. It seems to work well, but you know, you're talking about moving large sheets or large rolls of mesh around, so that could be problematic. This national park worker is she's removing individual stalks one at a time with a pair of clippers. So that's another option, is to mechanically cut them and to remove them. There's some kind of disagreement, I would say, which would probably be the theme for knotweed, about whether or not the cut stems can regrow roots. Randy Prostak, who's the UMass Extension and weed specialist, he said he's done a bunch of experiments using, just trying the leaves, trying the stems, and the only thing he could get to root was the rhizome, the actual root structure itself, and that you only need a small amount. About an inch is enough for it to start a whole new plant. But he said the only way he could get the stems to root was to use rooting hormone. So, that maybe it's a concern, but erring on the side of caution, cutting, removing, putting them in a spot to desiccate so that they can't get back into the soil is definitely not a bad idea. And this kind of conflict again, that leads to different extension agencies recommending different treatments. So UMass Extension is recommending mowing as part of an integrated pest weed management approach. Missouri State Extension is saying, please don't mow it. And the thought there is that when you mow, it does kind of knock back the growth, but it causes it to grow laterally, and it'll kind of send runners to come up. And so you're reducing the abundance, like the above ground abundance, right where you're mowing, but you might be increasing the lateral growth as well. So that's something to think about. And then kind of the last method that we have, well, actually there's two more methods, sorry, is cultural control. This is something we talk a lot about in agriculture, but is appropriate for knotweeds as well. And that, the biggest thing that we can do to stop the spread of knotweed is to limit the spread of infested topsoil. So because the rhizomes, you just need about an inch, as long as it's got a node on it. If it's in the soil and it gets watered, it'll send up a shoot. So, in the UK, what they do as part of this remediation process is they remove the first couple of feet of topsoil, they sift it. And so they're going through, and it's incredibly labor intensive, and they're trying to remove any of the rhizomes from the topsoil so that it could be treated and then reused again, potentially. But it is kind of treated like a hazardous waste almost. And just for fun, culinary, a lot of people, when you say knotweed, they actually will tell you that they've eaten it. And it is related to rhubarb. So you can pretty much replace knotweed for rhubarb in almost any recipe. I haven't eaten it myself, but I am curious and interested in trying. These are from Bon Appetit, so they must be pretty good. But you've got a crispy rabbit pickled knotweed on the left. There's almond cakes, knotweed on the right. And a colleague of mine in Japan, he says what they do is that they take the leaves, especially the larger leaves, and they steam them, and they kind of do like a folded grape leaf. And he really likes that. So you can never, so culinary is often promoted. It's not, you can never eat enough of it to really put a dent into the population, but at least it can be fun to be like, okay, it's not the end of the world, let's get rid of it, but maybe I'll try a little crispy rabbit in the process. So back to how to integrate this, and we've got chemical controls, we've got mechanical controls, and I really like this kind of suggestion from the Penn State extension, where we've got knotweed coming up in March and April, and to hold off and not to do anything right away. Let it grow. And what's happening is it's sucking out. It's got this big rhizome that stored all this nutrients up over the winter, and that's why it shoots up in March. And it's just right away it's come up because it's got all this stored energy ready to go. So let it use that energy, send up the shoots. It's going to get tall, but it's going to use up a lot of energy from that rhizome, and that's where you can come in early June, end of May, and cut it down. Remove that those cuttings so you're not accidentally making the problem worse. And then it hasn't had a chance to, or the thought is that it hasn't had a chance to replenish the rhizome yet. So it's still got energy, and it's going to send up another round of shoots. But at least you've kind of knocked back a bunch of its growth potential. Now the second round is going to come up, and that's where you can use an herbicide or a second cutting if you wanted to. You could keep applying you could keep going with the cutting treatments, just so that you're knocking the rhizome down each time. It probably will take more than a single season to make for it to disappear, but at least you can really start to make a difference in the size of the rhizomes and the amount of energy that they've got, and that might help some of the native plants emerge and establish in the meantime. So I like this timeline. And you might find other similar timelines online as well. But what we're really interested in is biological control. So we're hoping to work with folks on Nantucket to release the Psyllid Aphalara itadori. We've been doing work in Western Massachusetts, and this is part of a team in North America and actually an international team trying to introduce biological control agents for knotweed species. It was just recently approved in 2020 for North America, and we've got a few different strains. So it's funny, we've got this insect right here, and it's now because we're calling it a biocontrol agent, we kind of apply like FDA medical terminology to it. So this is this Kyushu strain of Aphalara itadori. We have a few other strains. There's the Hokkaido strain, and there's also a new strain, the Murakami strain, and each one of these species preferentially feeds on a different species of knotweed. Some of them, like the Kyushu strain, can feed both on Japanese, its preferred host, and the Bohemian knotweed. The Hokkaido strain is really quite specific just to giant knotweed. So that's more being targeted right now in the Pacific Northwest. But the Murakami strain, because it's feeding on the hybrid, or it's preferentially feeding on the hybrid, it does seem to be a little bit of a mix of the two, and it will use the other hosts. So that's really quite promising. And that's being released right now in the UK and the Netherlands, and they're having some fairly good establishment. But it's still going through the review process here in the United States. So that's hopefully coming up soon. But maybe you're wondering, what is biocontrol or biological control? It's a term that that gets used quite frequently, and kind of textbook definition that I like is the promotion of natural enemies. These can be predators, they could be herbivores in the case for the knotweed project. They could be a pathogen, and the goal is to reduce the abundance of a target pest or weed. And it's for centuries it was pest management. That was the only option. You promoted what was available, the natural enemies that were found in your garden or found in your region, to feed upon the potential pest species. We have documented evidence from the 3rd century Common Era in China, where growers were, in the textbooks, it talks about how to promote the ants in your citrus orchard so that they'll feed on the pests there. So this is longstanding. In the US, it really kind of took off in the 1880s when this beautiful beetle here was introduced to feed on cottony cushion scale. I was gonna say there's this great book that you should read called How to Make a Spotless Orange, and then I looked online and it's like $75 used on Amazon, so you can get it from the library. But it goes into kind of the history of how this biological control program was started. It was the first formal one in the United States, and it goes into the USDA and the California Department of Food and Agriculture, how they looked in Australia for a predator that could feed on this pest, which was wiping out the citrus industry. And it's why you get Florida orange juice, and the oranges that you eat are primarily from California. It goes back to this biocontrol program. Today, we call this classical. Some people are starting to call it introduction biological control. And the idea is that we're reuniting our pest, our non-native invasive pest, with a natural enemy from the region of origin, which is slightly different than what the Chinese farmers were doing historically, which was promoting, and then we call that conservation biocontrol. And just because this is a weed talk, if I were asked to say the most famous example, it would be this one, the biological control of floating fern. This was an incredible success. The bottom left and the bottom right photos, this is the same. I mean, I guess there's a little bit of a difference in the mountain, but it's supposed to be the exact same reservoir in Australia. And on the left is that reservoir infested with floating fern. And the problem is that it makes the reservoir no longer a viable water resource for the population. It clogs the equipment. The water needs to be retreated. So this was a real problem, and it was a real problem in much of the developing world where this floating fern got spread to. In 1978, the Salvinia weevil, which is this beautiful weevil in the top right, was introduced. And within 15 months, this is the before and after photo. So this is kind of like the promise of biological control, is that a natural enemy, once it's reintroduced, or once it's, in this case, introduced for the first time into a new habitat, kind of brings it back to what it was like in the pest's native range, where it's generally not considered a pest. And it was a 50,000-fold reduction in the dried biomass of floating fern in this single reservoir. So that's kind of what we're hoping for knotweed, is that we can introduce an agent like Aphalara itadori that will knock back the knotweed and kind of make it so that we don't have to go out and do chemical control. We don't have to go out and mow it. It can just be part of our normal kind of land management. And this is a project that I've been involved with for only recently, but it started in 2003. Collaboration between Dr. Richard Shaw at CABI in the UK, Dr. Robert Boucher from Agriculture Agri-Food Canada, and Dr. Fritzi Grefstadt at Oregon State University. Together, they screened both in the literature and field surveys with their colleagues in Japan, 180 potential insects to be introduced. And at the end of these screenings, the only one that they felt was comfortable for testing was Aphalara itadori, the psyllid. And this is because of the concerns of an introduced insect feeding on an alternative host. There's a really stringent process by which, again, if we want to talk about the FDA, there's a review that has to be done where you have to show that it only feeds on the target host. And you compare it to closely related species. And so here's a very not the most beautiful slide, but it's a table of just, and this goes on for several pages if you want to look at the manuscript, of all the different hosts that they tried to feed the psyllid upon. And it really does only feed on a Japanese knotweed, Bohemian knotweed, and another species found in Japan as well. So none of the North American species had any type of egg laying. There was no signs of feeding damage. So this was really exciting for the team. But they started the project in 2003, and it took until 2020 for it to be released. So this is definitely something, it is a slow process. It's a methodical process because you don't want to make mistakes. And so we're testing as many different species as possible for the feeding, just to limit that kind of non-target host impacts. And then, so this is where we come in 2020. Joe Elkington, pictured here, and this is, I don't know, for folks who were involved with the Black Oak Gall Wasp project, Cameron Friedman Smith. Together they're collecting Black Oak galls in probably right around the field station site. But this is part of a multi-regional project that Dr. Grevstad has organized. The lab with Joe Elkington, we're the Massachusetts aspect of it, but there's folks in Washington State, Oregon, North Carolina, New York, Rhode Island, and then us. And we're all trying to introduce the psyllid and study it at the same time. And so what we decided to do was a little different than some of our colleagues have done, in that Joe was really adamant about, we want to make a positive change, but we also want to do good science in the process. And what can we learn about the insects, rather than just letting them go and seeing what happens, which is great. I mean, we want to reduce knotweed, but what can we learn in the process? So what we decided to do was, rather than just releasing the psyllids, we brought them into the greenhouse and we reared them so that we could have even more individuals to release. And we looked at a variety of different habitats that are kind of common, at least here in Western Massachusetts, for knotweed. In each location, so this top left corner, we did paired releases where we put psyllids in the bags. We left the bags on for 2 weeks, and then we randomly opened one of the pair to see what would happen, whether there'd be predation, whether there'd be dispersal, and from there. So, this is common in roadsides. It's common in meadows, and that's been interesting places to work. Forest understory definitely has lots of knotweed. Riparian, of course, and parking lots. So this is the UMass campus photo right there. You can see the buses, and it's just lined up along the parking areas. It loves disturbed habitats. And there's actually landscaping fabric that it's growing through, which is I love, because I've tried to dig through it. But here we are. We brought the knotweed plants. We had to get permits from MDAR, Massachusetts Department of Agricultural Resources, to grow knotweed, which it's good. It's good to have permits. It seems so funny to have these giant knotweed stands, large knotweed stands. They're Japanese knotweed, right outside the door. And then I've got my permits to have them in the greenhouse. But they're very healthy plants. They grow wonderfully. Even I, who do not have a green thumb, can grow knotweed. It's fantastic. And after about a month, we went from the initial shipment that we got from Dr. Grevstad to what you can see here. Each one of these is an individual adult psyllid. You can see, if you squint really hard, along the main stem, there's these kind of like smaller circles. Those are the immature stages. And so pretty much we got fruit fly abundance within a month, which was really exciting. And we ran into one hiccup. This is my field assistant, Wesley. He is now 2.5, but at the time he was a little bit younger. We, because of COVID, ran into some greenhouse access restrictions, and also we had this just really early season heat wave, which at the time I thought was shocking, but based on the other heat wave we just had this weekend, it seems maybe the new normal. But anyway, we had to move out of the greenhouse. We set up under my deck, and here Wes is working with his sandbox while I'm counting psyllids on the side. But we had enough psyllids so we could do these paired releases. And we've got some really clear results, which is always nice. They really seem to be doing better in the sleeves. So this could be predation, this could be dispersal, and that's something we're trying to work on. But it looks like if we want to get them established, starting them and potentially leaving them in the sleeves for the growing season. Seems to be a really effective way to keep the population stable. We also tried field cages, and I promise this, afterwards, I did close that field cage with a clip. It was just, this was the photo as I was getting it set up. Here's Joe Elkington, and in the little bug dorm here, the black dots along the top, those are all psyllids. They're really jumpy is the description for psyllids in general. They do like to bounce around and move, and so they're just waiting to disperse on the top of the container there. We've got them in the field releases, and this is what it looks like in a field cage. Again, we've kind of got this fruit fly abundance, but they just disappeared. After a month, they were gone. And we assume that is due to predation, particularly from ants, is what they're finding in Canada. So hopefully, we took this information, we came back for a second year of study, and I had to dust off my home ec work. It's been a while, but I made these 2 meter tall mesh bags to cover a fully grown knotweed plants, and we left them on for the entire season. We put about 100 psyllids in each of the bags. And we let them there, left them there. We looked at them throughout the season, and we also did some more open air releases. But kind of striking, we also found with our open air releases last year psyllids throughout the summer. So in our field cages, they just disappeared in 2020, but 2021 they persisted. So maybe I don't know. I'm not the best animal care person, so maybe we've bred for some hardy ones, but it looks like we're going in the right direction at least. And just a few days ago, I went to survey our sites, and I found 13 adults, which I was shocked. I was not expecting to find a natural occurring population. And I talked to Fritzi Grefstad. She's found a couple of adults overwintering in North Carolina. Lisa Tewksbury at Rhode Island, they've also found a couple individuals. So this is the first year that we have survival of kind of natural populations of the psyllid in the Northeast or the East Coast. So I'm really excited that this is the trajectory we're going to go. And that brings us to Nantucket. And I'm sorry for being a bit long-winded about it, but these are two of the local magazines, newspapers that I know well. And this knotweed's a problem. It's a problem in many habitats. You'll see it growing up along the sides of the bike path. I saw it on Pulpis bike path and going out towards Cisco. And this Lily Pond, this is a location where it's really quite problematic. And I know the folks at the land bank are interested in trying to control that population there. So maybe Nantucket could be a good location for biological control. And a couple of reasons I think that's the case is that we're getting some establishment in Western Massachusetts, but in the greenhouse, these things grow like fruit flies. Like, it's amazing. You just get hundreds and hundreds of them. A female will lay 30 eggs a day. She'll live for over a month as an adult. So I mean, that's incredible growth potential. But in the field, we were kind of seeing persistence as opposed to exponential growth. And I think this might be a little bit of a climate mismatch. This is an analysis we just published. But if you go back to Japan and you compare the climate of Eastern North America, Kyushu, which is the main southern island, which I've highlighted here, is really, this is a heat map, I'm sorry. So yellow means a really good climate match. Blue and the darker blue colors mean not so great, but at least something. No color means there's no predicted climate match. So Kyushu is actually not predicted to be a good source location for this psyllid, whereas Hokkaido and Murakami both seem to be pretty promising. So that's another reason we're interested in this Murakami strain. But I think the problem there is that, or the problem that we're having is that it's just too hot in Western Massachusetts. Like I'm sweating in my office right now, and it's probably, I don't know what it is, but it's hot already. 30 degrees is kind of an estimated thermal tolerance for this species, at least in lab studies. They stop laying eggs, you have increased mortality, and we frequently see 30 degrees Celsius, and I forget what that is. It's actually not that high. It's something like 84 degrees Fahrenheit. So that's a temperature that we get in the summer quite frequently. And so I think it's just a little too hot out here. But Nantucket, because it's moderated by the coastal climate, could be a really good location to get establishment. And the other thing is because of the moisture from the fog, from the rain, that could really help. And the location that we've got our overwintering population, that was a really nice forested setting with a stream running through it. And I think it just kind of kept it cooler and it kept it moist, which is two things which, I think Nantucket would really be a good source or a good location for establishment. And why else Nantucket? Well, this is a landscape level problem, and that's where biological control is particularly useful. When it's not just a farmer dealing with their crop, but when it's something that's occurring over vast areas, or multiple agencies are trying to work together, multiple landowners, biological control can provide that kind of landscape level control. Now, again, we've got the kind of moderated temperatures, both winter and summer, which I think will really help establishment. Mechanical and chemical control exists. It is possible, but it's incredibly time consuming and it's incredibly expensive as a result. So if this could help reduce the abundance of knotweeds, or at least reduce the biomass, that could make control easier with other methods, or allow the native plants to establish and kind of outcompete the knotweed stands. So a part of this, our next steps, I'm here today saying hi and fielding questions, but we've been talking with the Nantucket Conservation Foundation, with the land bank, just to see where's knotweed present, where are you concerned about it. Really interested in starting a dialogue with the public to see, what are best practices in your mind, or what are potential concerns that you have for introducing the psyllid. Hopefully we'll have a choice between strains of the psyllid, and particularly if we can use the Murakami strain, I think that would be ideal for Nantucket, where there is a lot of Bohemian knotweed, and so that might help establishment as well. And want to take it slow. I mean, maybe we can get out there this fall, or maybe next spring, but really it's better to kind of just take our time, work through the details, and make sure that we're introducing the right insect at the right time and the right places. So definitely don't want people to feel rushed into this. And just at the last second, I was thinking, maybe this also could be a fun way to collaborate with the local school and the Nantucket High School students or biology students who are interested. It's easy to find knotweed, and the psyllid, it pops right out. So it could be a fun way to get students involved in a research project. So I'll provide the PDF. Hopefully that's easy to share. But if you're interested in the biological control of knotweeds, there's a Forest Service pamphlet that it has a lot of great information, proper survey techniques, the history of the program. This very long resource, I believe, is from the Conservation Foundation, Nantucket, about their efforts to remove knotweed. And again, I mentioned Dr. Randall Prostak here at UMass Extension. His slides as part of last year's webinar, where it does go into which formulations of glyphosate, for example, are appropriate in which settings. But that's something that you definitely want to make sure you're a licensed applicator or have the appropriate license for the location. With that, thank you, Yvonne, so much. I'm sorry I took up a lot more time than I had intended. Thank you to Kelly and Karen at the Conservation Foundation, Guthrie and Rachel at the Land Bank, the Elkington Lab, of course. Jackson, Christopher Callahan was an amazing undergrad who did most of the field surveys for us. And Roy Van Drysch, who's retired now, but he can't quite kick the habit. So he comes in almost weekly to make sure that the project is still going forward. Fritzi Grefstad, of course, for supplying and for all of the legwork she's put into this. The UMass greenhouse staff for letting us grow knotweeds which was funny, but anyway, thank you so much. And I would love any questions that people have, and thank you, Yvonne.
Thank you so much. That was really wonderful. We do have a few questions. It's up to you, Jeremy, if you'd like to leave your slides up or take them down. You can do whatever you feel you want to do.
I can leave them up, I guess.
Up to you. If you need to refer to any, you can either share them or leave them up. But let's do this. Let's keep you up here. Yeah, let's try that. Okay, so we have some comments and some questions. Catherine Howard in the Q&A box asked, My city removed a dirt pile from a park. The pile has been infested with Japanese knotweed. The city plans to use the dirt to fill under road work. Do you think that would be okay under heavy layers of asphalt, or should they sift the soil first?
Right. I guess I would be concerned, or I would be worried. I don't know how many layers of asphalt we're talking about here. I mean, you could very easily smother it with enough biomass. I do know that people are trying to put down the kind of plastic layers, like is being for agriculture, to try to smother it. I don't know if that's effective yet, but I do know that it's something that people are looking into. I would definitely follow up and just ask them that they've got a plan first off, and just make sure that they know that the rhizomes are quite hardy, yeah, I would want to follow up with them about that. Would be my answer.
Okay, Monique Fillon asks, do we know yet if other animals prey on psyllids?
Yeah, well, we do for sure. Definitely species of ants. We've seen ants, especially carpenter ants in the field. We've seen them on the plants. There's some smaller ants that I'm sorry, I'm not an ant expert, but we have a lot. It's knotweed and ants have a symbiotic relationship where the plants are providing extra floral nectaries to the ants, and it seems to be one reason maybe why there are not as many herbivores on the knotweed plants, is that the ants are guarding the plants in exchange for the resources. So we've watched ants just take a psyllid. Well, I've watched one, but spiders are generalist predators. They're common. So yeah, so we expect that they are getting they're a tasty meal for a lot of the local predators, and that was one reason why we did those predator exclusion studies.
Kelly Omond is asking, what amount of damage do the psyllids do to knotweed when well established, such as percent biomass reduction?
Yeah, I'm so sorry. So Fritzi Grevstad has a paper, and I forget the exact percentage, but I want to say it's in like the 15 to 25% range for biomass reduction. The two psyllids, she tested the Hokkaido strain on giant knotweed, and that was really quite, that was the greater of the two, and I'm so sorry, I forget the percentage, but it was a significant reduction in biomass. The other nice thing about the psyllid feeding on giant knotweed is it causes leaf curling, and I don't think I mentioned that. So the Japanese knotweed is mostly kind of like, it's sucking out the nutrients from the leaves, so the plant is less hardy. And that just kind of, it doesn't grow as large. But the giant knotweed, especially what the current thought is that the larval feeding on the kind of, I'm sorry, this is a knotweed leaf. It's coming up, and the psyllids are feeding on it. And just as it's supposed to open up and expand, but instead it does this, and it curls up. And that curling, the larvae actually crawl into, and they feed along that kind of central vein within the curl, and that kind of, it looks like it's sheltering them, but the psyllids are inducing it. So we don't know if it's maybe a pathogen or something, but for giant knotweed at least, there's not only the biomass reduction, but you can actually look at it and you can say, oh yeah, that plant is getting hammered right now. So that's kind of a twofer, which is nice.
Can I ask a follow-up to that? When you say this much percentage reduction, is it a reduction of the plant mass at the time that you put the psyllids on it, or is it a reduction compared to what you would expect it to be?
Yeah, and I'm so sorry. I meant to have that slide, and I just, as I was putting together, I forgot to include it. But what Fritzi did was she had paired, so control plants, and all the plants were grown in the same. They were the same, I believe they're the same cuttings from the same rhizome. so that there is no genetic diversity between the plants. And then they were grown in the same size potted containers. One treatment got the psyllids, and one treatment was remained without the psyllids, so that they would grow in the greenhouse, same light, same humidity, same everything. And then at the end, they dried it and they recorded biomass. And that's where they saw the reduction in the ones where the psyllids are feeding compared to the controls.
Another question. So how soon before we can get some of these in Boston?
Well, that's a great question. So we're starting small. I mean, they're all great questions, but we are starting small, and we're trying to move out. COVID really, if you saw, or if I could send you the original RFP that we have for this project, there's sites up and down in New Hampshire and Vermont, and we were gonna go crazy. And then with COVID, we had to kind of scale everything back. And we're like, well, we can do it locally. Let's start here and let's move out. We are going to work with Mass Audubon this season at some sites out here. And on my to-do list is to start talking to where they're excited about the research sites out here for potential releases. And I was hoping to follow up with them and do some other releases at some of the other Mass Audubon sites in the Boston area. So it could be as early as this season or potentially next season as well.
I have a question in the chat. There's a number of comments, but then there is a particular question that Eve Messing has posed, which is, you said you hope you have a choice of which particular strain of insect of the psyllid to use. Who decides which strain to use?
Right. Oh, well, that's a great question. So right now we've got for the tag review process. So this is done by USDA APHIS in collaboration with Fish and Wildlife, and then there's a public review period where they make a recommendation saying, we've got an insect, we think it'll be beneficial. Now let's get some public comment back, and that those actually are used, and that's one reason why it took from 2003 to 2020 for the approval, because there were concerns about the release. And so just, the scientists went back and then they worked through it. So right now there are two strains that have been approved by TAG for release, which are the Hokkaido strain and the Japanese, the Kyushu strain. So who decides which one to use? That's really right now up to the researchers or up to the, if we're going to do this project on Nantucket, it would be through discussion with the landowners, through the different conservation foundations, just to make sure that we're using the strain or we're doing something that everyone is comfortable with, because we don't, you can't get back goodwill. It's something that once it's gone, that's it. So we want to make sure we're doing it right. But the choice of the strain would really come down to what the local agencies and landowners are interested in using, and what's most appropriate, because the strains preferentially feed on different species. And I forget exactly which, the exact percentage of the species on Nantucket, but we would want to use the strain that would have the most impact by feeding on the most dominant species.
We have had one more question come in. How far can the psyllids disperse when they get a good population going? Can they move to take on other patches?
We hope so, and that's something that still is being still being studied. So I mean, the goal is that with a biological control program, with a classical biological control program like this, is that the psyllid becomes self-sustaining. It is both maintaining its numbers without us introducing more individuals, but it's also spreading through the landscape to feed upon every location. The group in the Netherlands right now, they are having a bit more luck with this Murakami strain, and so they are actually starting to look at dispersal distances, and seeing the abundance that they can find, and they're finding limited dispersal within a kilometer so far, but the numbers, they're still low, and like us, they're starting with these bag populations, and then they're kind of opening some of the bags. So the numbers are still quite small, but they are finding individuals dispersing about up to 1 kilometer right now. But historically, if you think about the Japanese archipelago, this is a species that can be found across the entire archipelago. So it can disperse across water in that case.
This is so interesting. I'm sure we could keep the discussion going. I did have a question. So you said these psyllids have been released in other countries. And in general, were there any negative, results from that at all?
So it is, to date, it has not been found on any non-target host. That is something that in the UK, where the first releases were done, in the Netherlands, where this more recently, the Murakami strain, and Canada, where there's been more introductions in those locations, and there haven't been any non-target hosts recorded so far. So we're really excited about that it is host specific. This, it is moving very slowly. So we this season was the first time. We've only had 2 seasons of releases, but the 13 individuals that we found at our study site, and the 2 individuals in Rhode Island and the 2 individuals in North Carolina, those are the, as far as we know it, like that's the 17 individuals that are wild in Eastern North America right now. So our goal right now is establishment, but of course, wanting to make sure that this does stay species specific, and that's why it took so long to do the review, and to make sure that this species wasn't going to cause problems. And this is, I said, you can supplement knotweed for rhubarb in your strawberry rhubarb pie if you'd like to, and that was actually a real concern, was that this would start feeding on rhubarb, and pretty much agricultural pests or agricultural items are non-starters for a biocontrol program. So that was one of the first filters for that 180 species, was anything that could feed on rhubarb was tossed out, and that the psyllid, it made it through that pass, and it's made it through all the other testing. So, I mean, it looks like a really promising and a really host-specific species. It's even host-specific within knotweed species. So that's something that we think is pretty encouraging.
I think, unless anyone else has a lingering question, I'll ask one more, and we can probably conclude. So if you have a successful release, as you see in these other countries, do you have to couple it, and maybe you said this, with other management techniques, or do you think this would be sort of the one control that would eventually take out the knotweed, or would you have to still go in and do a minor amount of physical removal?
I would assume it will be part of an integrated pest management option. I think it has the ability to reduce the abundance based on what we've seen in the lab, at least, and hopefully we'll see that soon in the field, or at least the biomass. But knotweed is it's such a vigorous plant, and it's hard to picture 6-8 foot tall plants just succumbing to the psyllid. What my hope is that the psyllid knocks down so if you've got a stand, and so my dream for this project is you've got the stand of knotweed, and right now it's a dense monoculture, and that's it. There's nothing else there. But by knocking it down 25%, all of a sudden you're giving native plants an opportunity to grow up, and then they can start competing with the knotweed, and between the native plants growing, the psyllid feeding on it, then maybe it'll be a minor plant that's kind of part of the community. With a biological control program, you never exterminate the item. You never exterminate the pest or the weed. What you try to do is reduce the abundance to where it's no longer a management concern. It would be like having poison ivy, which you're like, that's a bummer, but we can live with it. But yeah, so there was a question here that in the chat from Eve also about kind of bioremediation, and I have not heard of that. So that is something that I can definitely look into, and I can send a response if I find anything about it. But
Well, thank you so much. And yeah, we will make available any slides or materials that Jeremy sends to us. And this will be recorded and available on the Nantucket Field Station YouTube channel and shared with the Nantucket Conservation Foundation as well. So anyone who would like to watch this again or reach out to Jeremy with further questions. This has been really interesting. We thank you so much for your time and for sharing your expertise with us and posing this option, because it's really an interesting one. Thank you.
thank you, Yvonne, so much, and thank you, everyone. I can't see you, but